Welcome everyone. Thank you for being here today. It's my pleasure to share with you my poster presentation on computational resources for understanding the binding affinity of membrane protein-protein complexes and mutants. I'm Fathima doing my PhD under the guidance of Professor Michael Romiha from the Department of Biotechnology, Indian Institute of Technology, Madras, India. So coming to the introduction, as we all know, membrane proteins are a diverse class of proteins that resides within the lipid bilayer of the cell membrane. They execute wide range of cellular functions which include transport, enzymatic activity, signal transduction, etc. It is thus not surprising that they constitute approximately 1/3 of the cell proteome. They are targeted by nearly 60% of the marketed pharmaceutical drugs, and as you can see, among which GPCRs are the most prevalent drug target, followed by ion channels and receptors. Although numerous computational studies have been carried out on various aspects of membrane proteins such as structure, function, topology, stability, mutations, etc., however, large-scale investigations on binding affinity have not yet been reported. Since membrane proteins are thought to function in complexes, understanding the functions of these complexes is mainly dictated by their binding affinity. Experimentally, it can be determined using techniques like SPR, ITC, BLI, etc., and quantified using dissociation constant Kd and or the binding free energy or the delta G. It is also important to understand the mutational effect on the binding affinity and the relationship with disease-causing mutations, because definitely mutations affect their interactions, the binding affinity, as well as impair the critical functions and may lead to diseases. Mutations in membrane proteins have shown to be culprits across a broad range of human diseases such as cardiovascular diseases, cancers, etc. However, most research on binding affinity and mutation effects are focused on globular proteins, and currently no research is being done on membrane proteins. Hence, we decided to carry out this research. The rationale behind this study stems out basically from the recognition that there is a lack of readily available affinity data on membrane protein complexes. Secondly, factors influencing the affinity in membrane proteins is not completely explored, and also there is no specific method for affinity prediction in membrane proteins. So coming to my first work, which is MPAT, a database for the binding affinity of membrane protein-protein complexes and mutants. So despite there is an increase in experimental affinity data on membrane proteins in literature, they are dispersed, necessitating their compilation into a comprehensive database for further analysis. Hence, we developed this database named MPAT, and this is the overall workflow of the database. So we have collected the experimental affinity data of membrane proteins from PubMed, which is a literature source, and also from certain specific journal websites, using a set of keywords such as the names of membrane proteins, structural functional terms, experimental techniques, etc. And we also used a combination of these terms to obtain the relevant papers. And after manually reviewing each of these papers, we collected the following data details, and efforts were made to standardize all the units of the thermodynamic data to make the results comparable. In addition, we also integrated affinity data which is available in other existing databases which are for protein-protein complexes in general, and then manually collected the incomplete data and other necessary details missing in these databases. So MPAT has an easy-to-use interface, and we provide the options to build search query, display, sort, download, and upload the data. These are the features available to the users, and this is the homepage of the database, and currently we have 5376 entries, which includes both wild type and mutant data, and detailed statistics is available in our website. So this is the search page. So we have both simple search page where the user can search by keywords like protein names, PDB ID, etc., and obtain the results. Users can also search by entry ID available and retrieve all the information related to that specific ID. And in the advanced search page, you can see that users are provided a build search query form as well as a display column form, and user can build a query using combination of multiple search options, and the results will be displayed in a table form, and also you can download the results in an Excel file format. Coming to the next objective, which is basically MPA-PRED, a machine learning approach for predicting the binding affinity of membrane protein-protein complexes and membrane protein-protein complexes. So as I mentioned earlier, experimental techniques for determining the affinity of membrane protein complexes, they are actually expensive and time consuming, making them infeasible for large scale applications. So there is a huge demand for accurate computational approaches to determine the affinity of these complexes. So we developed this method called MPA-PRED. So this is the overall workflow. So we have collected the data set from MPAD again using the following conditions as described here. And our data set consists of 114 membrane protein complexes, and features we have calculated both structure and sequence based features as listed here. And we also classify the complexes based on functions, type of membrane proteins, complex type, etc. And finally, we have developed regression models for each of the class, and the performance was validated using Pearson correlation coefficient and the MAE, and also did the jackknife. And finally, we developed the MPA-PRED web server. So these are the results. So this is a scatter plot showing the relationship between the experimental and predicted affinity on the jackknife. So basically, our method is able to achieve a correlation of 0.9 and MAE of 0.72 on training, and a correlation of 0.83 and MAE of 0.91 on jackknife. And aromatic and charged residues at the interface were found to be an important feature in most of the classes. And we also validated the performance of our model on an external blind test set of 11 complexes and obtained an overall MAE of 0.87 kcal per mole. In addition, we also compared our method with existing affinity prediction methods, which are for protein complexes in general, which is like Prodigy and PPA-PRED. And as you can see by looking at the MAE, that our method performs better because a few reasons. One is that we have incorporated both structure and sequence based features, well compared to the other two, which is like Prodigy is a completely structure based method and PPA-PRED is a sequence based method. And we also did structural and functional classification, and which has indeed helped to improve the performance. And another reason is that our method is specific for membrane protein complexes. So this is an example of how to input and retrieve the data. So you have the predicted delta G and the calculated KD being displayed in the result page, and the server is easily accessible at the given URL. So in conclusion, we have basically developed the first and specific database called MPAD for the binding affinity of membrane protein complexes, and contains nearly 5,400 data from 950 proteins. And we also developed a novel sequence and structure based ML method for affinity prediction of membrane proteins, and also developed a web server for large scale analysis. So we anticipate that these resources will indeed help to provide insight into the structure and function of membrane proteins, and can help in the rational design of drugs and fostering advancement in drug design strategies. So these are the references, and this is my poster, and I cordially invite all of you to visit my poster, and I would be delighted to discuss my work with you, exchange ideas, and answer any question you have. I look forward to engaging in insightful conversations at my poster, and thank you.